I'm Xuyuan Xiao. Welcome to our lab. So research in our laboratory is focused on understanding how plants fight against fungal pathogens. Specifically, we have been trying to understand the molecular basis of a broad-spectrum resistance against powdery mildew. Powdery mildew is one of the most common and important plant diseases that are caused by obligate biotrophic fungal pathogens in the order of Eraciphes. The most exciting discoveries so far we made is that we have identified a plant resistance protein called RPW8 that is specifically targeted to the host pathogen interfacial membrane called the extrahostoral membrane, which represents the host pathogen interface.
My name is Harley King. I'm a fifth-year graduate student in the Shao lab. This plant here on the left, these are Arabidopsis plants, and they have been infected with powdery mildew. Whereas contrasting these plants here on the right, these plants are free of powdery mildew. These plants on the right have a special protein called RPW8.2. It's this protein that the Shao lab spends a lot of their time investigating. In fact, all of us here in the lab have projects that are tangentially related to RPW8.2, and my projects are no different. I'm investigating the role of phosphorylation in RPW8.2, specifically what happens if the protein is phosphorylated, and if it is phosphorylated, what happens after it's phosphorylated. Phosphorylation is when a phosphate group has been added to a specific portion of the protein. And it's these phosphate groups that give molecular clues to how the protein is to be trafficked, to where it's to go, and can ultimately hint at its function.
I'm Chong. I'm a third-year graduate student from the Shao lab, and I'm interested in studying the involvement of lipid signaling in plant defense. And specifically, I'm working on two phospholipases that may be involved in plant innate immunity. So specifically, phospholipases are a family of enzymes that use structural phospholipids. So right now I'm trying to understand in which signaling pathway these two phospholipases may be involved and how this signaling pathway affects other defense signaling pathways and how this signaling pathway affects RPW8 trafficking to the host pathogen interface.
So my name is Yi. I'm a postdoc in the lab for almost 3 years. So part of the reason why this gene, this RPW8, can confer such a great resistance is that it goes to a membrane that encasing a special fungal feeding structure inside plant cells called hostoria. So hostoria is an organ generated by the fungus inside plant cells that can help fungus to get nutrients from plants. So RPW8 goes to that membrane and it can activate on-site defense reactions against powdery mildew pathogens. So that's part of the reason that we understand that why it can cause better resistance against a whole series of fungal pathogens. So currently our lab is focused on understanding the further or the detailed molecular mechanism underlying these defense reactions triggered by RPW8. My projects are focused on the special membrane surrounding the hostoria, like the biochemical features of this membrane and proteins on the membrane and how these proteins are targeted to this membrane.
Based on our studies, we found RPW8 functions right at the host pathogen interface to constrain the fungal feeding structure called the hostorin. So in terms of defense action, RPW8 functions just like a molecular missile.
Hi, I'm Xianfeng Ma. I'm a postdoctor in Dr. Xuyuan Xiao's lab. Now I'm working on RPW8-related project supported from a grant by Gates Foundation. Now I'm quite interested in using RPW8 as a vector to target the other antimicrobial peptide to the host pathogen interface. I'm also working on a genetic screening project that aims to identify and characterize the cellular trafficking component that is required for targeting RPW to the host pathogen interface.
I am Jie Liu. I am a visiting scholar and come from China. I'm working on sugar transporter protein from Arabidopsis selena. I hope to find one or several sugar transporter protein manipulated by pulmonary mildew. Now I construct a YP fusion with sugar transporter protein. Commonly, the sugar transporter transports some sugar for Arabidopsis development and growth. But when Arabidopsis is infected by pulmonary mildew possibly control sugar transporter for its service.
So currently, our laboratory is trying to understand how plants have evolved such an exquisite defense system and how this system works. And we are also trying to utilize RPWA as a unique and powerful tool to understand the origin, biogenesis, and the molecular warfare at this host pathogen interface. And also, we want to engineer RPWA as a delivery vehicle to target all the antimicrobial proteins to the host pathogen interface, so that we can build up more powerful missiles to help plants fight against fungal pathogens.